Hello everyone, my name is Alicia, and together with my groupmates Xiaowen and Nicole, we'll be presenting on our bioinformatics project. So this is the table of contents. We'll be covering on the introduction and objectives, then we'll be speaking on the bioinformatics tools used, and lastly the analysis and conclusion. So for our gene of interest, we have chosen the human leukocyte antigen HLA gene. The HLA gene is a group of proteins that are made from the major histocompatibility complex MHC that enables the body to distinguish between proteins made from our body and foreign particles. There are three types of MHC class 1 genes, HLA-A, HLA-B, and HLA-C. They produce proteins that are present on the surface of cells. These proteins have been previously binded to a MHC1 molecule in the endoplasmic reticulum. The HLA gene is also highly polymorphic due to the changes in genetic sequence, such as the sharing of genetic materials between alleles, point mutations, and genetic drifts. Since HLA is expressed differently for each individual, HLA typing is essential to prevent rejection during blood or organ transplantation. In this slide, we show that we have used the National Center for Biotechnology Information website to obtain the FASTA protein sequences of the three main MHC class 1 genes, which are HLA-A, HLA-B, and HLA-C. The following slides will show the FASTA nucleotide or protein sequence for the MHC class 1 genes. This is the HLA FASTA sequence, HLA-B, and HLA-C. So for our objective is to find and compare the similarity of different HLA gene sequences within Homo sapiens and across different species, determining the suitability for transplantation. Now I'll pass the time on to Xiao.
Next, we'll talk about the different bioinformatic tools used to help us achieve the objective as mentioned by Alicia in the previous slide. First, we'll be finding the function of the HLA protein using Cav search. By using Cav search, we can determine the function of the three HLA protein sequence and classify their protein structures. After submitting the protein sequences to the three HLA genes to Cav, the functional families and the representative Cav domain is obtained as shown in the slides. The same functional family and representative Cav domain is obtained for all the three HLA protein sequences. Next, the profile and the pattern of the HLA protein sequences was determined using ScanProcyte. A pattern is a physical or qualitative expression of a motif. ScanProcyte is used as it is able to determine the patterns and profiles of the protein sequences and show the procyte pattern or consistent sequence. After submitting the three HLA protein sequences to ScanProcyte, the pattern PS00290 IgMHC immunoglobulin and major histocompatibility complex protein signature was obtained for both HLA-A and HLA-B. HLA-C, however, did not have a pattern. A further look into the IgMHC protein signature shows that it is a tetrametric immunoglobulin molecule made of light and heavy chains linked by disulfide bonds. There are two types of MHC molecules, class 1 and class 2, and these molecules are made of two chains. As seen in the slide, the main difference between two classes of MHC molecules are the number of extracellular domains present in their alpha and beta chains. It is also known that the Ig constant chain domains and a single extracellular domain in each type of MHC chains are related, and this category of Ig-related proteins can be detected through a small pattern around the C-terminal cysteine involved in a conserved intradomain disulfide bonding. The consensus pattern found in the protein sequence of HLA-A and HLA-B is shown in the slide, with the cysteine residue in the pattern involved in the disulfide bonding. To compare the similarity between the three different HLA sequences and the similarity across the different species, we'll be using T-Coffee as a multiple sequence alignment software to identify and determine the similarity between the different HLA sequences. UniProt-KB will then also be used to find the HLA protein sequences of the following species: monkey, pig, and mouse. The HLA protein sequences obtained will then be compared with the three human HLA sequences as part of HLA typing to determine which species is suitable for xenograft transplantation into humans. As mentioned, T-Coffee is used as a multiple sequence alignment tool to compare and identify the conserved regions between many protein sequences. In this case, pairwise sequence alignment is not used as it is too weak to identify the similarities among distantly related species unlike MSA. By using T-Coffee, The degree of similarity between the amino acids present in a particular position of the sequence will be highlighted with a different color, such as red for the most similar conserved regions between the sequences. T-coffee also provides a score for the sequences compared, showing a percentage of how similar the sequences are. As seen from the results, after comparing the 3 human HLA protein sequences in T-coffee, the sequences are mostly similar and consist of large amounts of conserved regions highlighted in red. The score obtained was also 99. To determine whether some common species are suitable for xenographic transplantation into humans, the HLA sequences of pig, mouse, and monkey were determined using UniProt KB. This slide shows the HLA protein sequence for a pig. After comparing the 3 HLA sequences with the pig HLA protein sequence, it can be seen that the pig HLA sequence is mostly similar to the 3 human HLA sequences, as the sequences are largely highlighted in red, with a few spots highlighted in yellow and green. A score of 93 was also obtained. The mouse HLA protein sequences was also obtained using UniProt KB next. As seen from the results, after comparing the 3 human HLA sequences with the mouse HLA protein sequence, a score of 92 was obtained. Most of the sequences are conserved regions highlighted in red. However, as compared to the MSA results with the pig HLA protein sequence, more of the regions were also highlighted in yellow and green, showing that the mouse is less suitable as a donor for humans as compared to a pig. Lastly, the monkey's HLA protein sequence was also determined using UniProt KB. As shown in this slide, the score obtained for the MSA between the 3 human HLA protein sequences and the monkey's HLA protein sequence was 95, the highest among the 3 different animal species we used. It can be seen that the HLA sequences of the humans and monkeys are mostly similar and largely conserved from the red highlights throughout the sequence, with very little portions highlighted in yellow. Now I'll pass on the time to Nico, who will be discussing on the analysis of the results obtained.
Now I will be explaining our results and analysis obtained from the MSA results. Firstly, we will analyze the MSA results of the 3 human HLA protein sequences obtained from T-coffee. From the MSA results, the 3 HLA sequences are very similar to each other. A score of 99 is obtained, and most of the sequences are highlighted red. This emphasizes that the HLA sequences are very similar to each other. However, even though the sequences are highly similar, HLA typing is still needed to be performed, as the human HLA gene is highly polymorphic. Hence, this is to ensure there will be no rejection by the recipient due to the different HLA sequences. A recipient can opt for a xenograft for organ transplantation, which is when the tissue or organ is retrieved from a different species. UniProt KB is used to obtain the different HLA sequences from different species: monkey, pig, and mouse. The monkey HLA sequence is the most similar to the human HLA sequence in terms of the score obtained of 95, followed by pig with a score of 93, and lastly mouse with a score of 92. Hence, we can assume that monkey may be the most suitable to be used for a xenograft compared to pig and mouse. However, HLA typing is still required to determine the suitability and compatibility of the grafts. In conclusion, we can conclude that monkey is the most suitable among pig and mouse to be used as graft, as its HLA sequence is the most similar to the HLA sequence present in humans. With the help of different bioinformatic tools such as NCBI, UniProt KB, T-coffee, and ProCyte, we are able to find out more about our gene of interest, the HLA gene. We are also able to make a comparison on the similarity between the different HLA sequences among different species. Using the MSA tool from T-coffee, we are able to find which species is the most compatible to be used as graft, and the most suitable species is monkey after our analysis and results are obtained. Here are our references. Thank you.